If you look at the global health picture, cardiovascular disease is still #1, the leading killer worldwide.
Absolutely. It's a huge burden.
And for decades, so much of what we thought we knew about how the human heart works and, well, how it fails, it came from animal models.
And there's the rub, right? That's the core problem we keep hitting.
Yeah, because those models, they often just don't translate. What works in a mouse doesn't necessarily help a human patient. They miss something.
I miss a lot, actually, especially the complexity. The human heart isn't just muscle cells pumping away in isolation. It's a whole ecosystem.
An ecosystem, I like that.
Yeah, and the existing models are really missing key parts of that ecosystem, particularly the interaction, that constant crosstalk between the heart muscle and the immune system.
The immune connection.
It's vital. And it's often ground zero for where diseases, especially chronic ones, can really take root.
Okay, so that sets the stage perfectly for what we're diving into today. We're looking at sources detailing work led by Colin O'Hearn and Ada Aguirre, and they tackled this exact translational gap, this missing immune piece, by building something really quite new. The human heart macrophage assembloid. We'll probably call it the hHMA to keep it short.
Yeah, the hHMA. It's essentially a next-gen heart organoid. Think of it as heart organoid 2.0.
Okay.
Because it integrates a very specific and very essential type of immune cell right into the developing heart tissue structure. So our mission today, for you listening, is to unpack how this platform really mimics those crucial immune cardiac interactions.
Giving us a window we haven't really had before.
Exactly. Unprecedented insight, really, into both how the heart develops normally and also into disease mechanisms. Specifically, we'll get into inflammation-driven atrial arrhythmias later on.
All right, let's start at the beginning. How do you actually build one of these hHMAs? What's the foundation?
So the starting point is the standard human heart organoid, the HHO. Researchers have gotten pretty good at growing these.
Right, those have been around for a bit.
Yeah, and they do replicate a lot of the basic anatomy. You get different heart muscle cells like ventricular and atrial cardiomyocytes. You get cardiac fibroblasts, the structural cells, endothelial cells. It's a complex mix.
There was always something missing. Some key residents weren't showing up to the party.
Precisely. And the key missing residents are the star of this particular show. Tissue-resident macrophages, or TRMPs.
Okay, TRMPs. So these aren't your typical immune cells floating around in the blood.
Not at all. These are highly specialized. They actually originate from the yolk sac very, very early in embryonic development. They migrate to the developing heart and basically set up shop permanently. They live there.
Wow, okay, so they're true residents. Why does the heart need these living immune cells so early on? What's their job description during development?
Well, we know from animal studies that they are absolutely essential. You could almost say non-negotiable for the heart to form correctly.
Non-negotiable, huh?
Yeah. They influence pretty much everything. Building new blood vessels, that's angiogenesis, remodeling the tissue as it grows, and maybe surprisingly, they even play a role in setting up the heart's electrical conduction system.
So a heart trying to develop without these TRMPs is fundamentally flawed from the start.
That's the picture we get, yes. It would be seriously impaired. And since the standard hHOs, the heart organoids, didn't just make these TRMPs on their own.
They had to be put there, engineered in.
Exactly. They had to figure out how to add them.
Okay, but adding cells is one thing. Making them feel at home, making them actually integrate and act like residents, not just visitors passing through. That sounds tricky. How did they manage that?
That was the clever part. The strategy was all about mimicking natural development, developmental mimicry.
Okay.
So they first generated monocytes, a type of immune cell precursor from human pluripotent stem cells, HPSCs. But crucially, they tailored these monocytes to be just like the ones that seed the heart in an embryo.
How do you tailor a monocyte?
By its markers, its surface proteins. They selected monocytes that were CD14 positive, which is typical, but, and this is key, they lacked the CCR2 marker.
No CCR2. Why is that important?
Because that specific signature, CD14 positive, CCR2 negative, matches the known phenotype of the yolk sac-derived precursors that actually become these cardiac TRMPs in vivo.
Got it. So they made the right type of cell, but you also mentioned timing.
Oh, timing is everything in development. They looked at the timelines in mouse and human embryos. In the standard hHO process, the structure called the proepicardial organ gets induced around day 7.
Okay.
And researchers knew that in vivo, TRMPs start seeding the heart just around the time the heartbeat begins, which is shortly after that epicardial stage. So
they added their engineered monocytes When?
Right on cue. They started adding them at day 8 of the HHO differentiation process, just one day after that pro-epicardial induction.
And did it work? Did the cells stick around?
It worked beautifully. The integration was remarkably successful. These monocytes didn't just wash away, they actively integrated into the heart tissue, and they stayed there long term.
How long are we talking?
They tracked them, and the TRMP population stabilized right within the expected physiological range, about 1% to 3% of all the cells in the organoid. That's similar to what you'd see in a developing human heart. And they confirmed these cells were still present and integrated all the way out to day 60.
60 days. That's quite stable for an organoid system.
It really is. And it confirms that this hHMA platform is, well, basically the first robust in vitro model that successfully includes this critical step of human embryonic macrophage integration into the heart.
Okay, that persistence is huge. It means the model isn't just a fleeting snapshot. They've actually established that immune context. So now the big question, what are these TRMPs doing in there? What's their job? The hHMA model let the researchers basically write their job description.
Yeah, they pinned down 3 major roles. First up, they function as the electrician.
The electrician. Okay, intriguing. How so?
They found that the TRMPs are physically wired into the heart's electrical network. They form direct connections, called gap junctions, with the cardiomyocytes, the CMPs, the actual heart muscle cells.
Wow, physical connections. How did they see that?
They used immunofluorescence to look for a specific protein called connexin 43, or CX43. CX43 is the main protein that makes up these electrical gap junctions, and they found CX43 right at the contact points between TRMPs and CMs.
So like the insulation being stripped back and the wires touching.
Kind of, yeah. And it wasn't just a few connections they found. Over 85% of the TRMPs showed these direct CX43 links to the heart muscle cells.
That's right, 85%. That's significant.
Yeah.
But just because they're touching via CX43, does that automatically mean electricity is flowing between them? Does it mean functional coupling?
Great question. That's exactly what they tested next. They used live cell imaging with special dyes that light up when calcium levels change or when there's an electrical action potential.
Real-time electrical activity.
Exactly, and they saw that the integrated TRMPs had synchronized calcium spikes and action potentials right alongside the connected cardiomyocytes. They were firing together. So, yes, confirmed functional coupling. They are active players in the heart's rhythm.
Okay, this is where it gets really wild for me. If they're plugged into the grid, are they just passively conducting the signal from the muscle cells? Or do they have their own electrical personality, so to speak?
They absolutely have their own personality. This was a surprising finding. When they measured the action potentials, the APs, specifically within the TRMPs themselves, they found the TRMPs had uniquely prolonged action potential durations. Specifically, the APD90, APD50, and APD30 values these measures of how long the electrical signal lasts were significantly longer in TRMPs compared to the cardiomyocytes.
Longer APDs, so they hold on to the electrical charge longer. What does that imply?
It implies they're not just passive wires. This distinct electrical signature suggests TRMPs are actively modulating or influencing the heart's rhythm. That's a completely new role for these immune cells we're seeing here. A major discovery, really.
Okay, mine's slightly blown on the electrician front. Let's move to the next role. You called it the cleanup crew. This sounds more like traditional macrophage work.
It is, but seeing it happen in situ in a human heart model is key. This role focuses on efferocytosis.
Efferocytosis. Fancy word for
clearing away dead or dying cells, apoptotic cells, and other cellular junk. It's crucial for maintaining healthy tissue and preventing inflammation that can be triggered by debris just lying around.
And the hHMA model showed the TRMPs doing this.
Very effectively, yes. They were actively engulfing apoptotic material within the organoid.
Is it just a general vacuuming process, or is it more targeted? Like, do they have specific tools for this cleanup job?
Oh, it's highly specific. They showed it relies heavily on a particular receptor on the macrophage surface called MERTK. That's ME proto-oncogene tyrosine kinase.
MERTK.
Think of MERTK as the sensor and the grabber mechanism for the apoptotic cells. Because when they used an inhibitor to block MERTK function...
Let me guess, the cleanup stopped.
Pretty much. The TRMP's ability to engulf those dying cells was significantly reduced. They also found out what eat me signal the TRMPs were looking for on the dying cells. It's a protein called calreticulin, or CALR.
Okay, so MERTK is the vacuum, CALR is the kick me sign on the trash. That's really specific.
It is, and identifying MERTK's role here is important. If you could potentially modulate MERTK activity, maybe you could influence how debris is cleared or not cleared in certain heart conditions. It points towards therapeutic ideas.
Right. Okay, electrician, cleanup crew. What's the third job?
The third role is the engineer. This covers their influence on the heart's structure and communication.
The engineer. So, shaping the tissue.
Exactly. They found that just having the TRMPs integrated actually changed the overall mix of other cells in the organoid. Their presence seemed to encourage the growth, the expansion of cardiac fibroblasts, the structural cells, and ventricular cardiomyocytes, the main pumping cells.
So they're like conductors, guiding the orchestra of cell types. Did they also affect how well the muscle cells work?
They did. They actually boosted the function of those ventricular cardiomyocytes. The hHMAs with TRMPs showed larger contraction amplitudes, stronger beats, and the muscle fibers themselves, the sarcomeres, were shorter, which is a sign of better maturity and contractile efficiency.
Stronger, more mature muscle. That's a big deal. What about communication? How do they talk to other cells?
They act like little communication hubs. They found TRMPs actively reprogram the local signaling environment. One key way they do this is by secreting extracellular vesicles, EVs, these tiny membrane bubbles packed with signaling molecules.
Little messenger.
Precisely. And specifically, they found TRMPs release SPLP1 positive EVs. When they analyzed what was inside these EVs, they found unique proteins related to things like endosomal transport, how cells stick to their surroundings, cell matrix adhesion. Basically, complex messages sent out to influence all the other cardiac cell types.
Okay, influencing cell populations, function, and communication. What about the physical structure, the scaffolding of the heart tissue?
They engineer that too. They directly regulate the extracellular matrix, or ECM. Specifically, they influenced how collagen type I alpha 1, COL1A1, was laid down.
Collagen, the main structural protein. How did they change it?
In the hHMAs with TRMPs, the collagen organization was more uniform, more ordered. The researchers described it as having reduced entropy, less randomness, less chaos in the structural layout. So the TRMPs help build a better organized, more stable tissue environment.
Wow. Okay, let's just recap that. These resident macrophages, the TRMPs, aren't just passive immune bystanders. In this human model, they're acting as integrated electricians modulating rhythm, cleanup crew performing targeted efferocytosis via RTKK, and engineers shaping cell populations, boosting function, sending signals via EVs, and organizing the ECM. It's quite a portfolio.
It really is. It paints a much richer picture of their indispensable role in the normal heart.
Which brings us to the next critical step. Can this more complete model, the hHMA, actually help us understand disease better? Specifically, they used it to model atrial fibrillation, AF.
Right. AF is the most common heart rhythm disorder, and there's a growing understanding that chronic inflammation plays a big role in driving it, but the mechanisms aren't fully clear, especially the immune contribution.
So how did they mimic that chronic inflammation in a dish?
They took mature hHMAs, let them develop for about 30 days, and then exposed them to what they called a chronic inflammation cocktail.
Sounds potent. What was in it?
A mix of known pro-inflammatory triggers. Lipopolysaccharide, LPS, which mimics bacterial infection, plus two powerful cytokines, interleukin-1, IL-1, and interferon, IF-endel. And they didn't just give a quick pulse, they exposed the hHMAs to this cocktail continuously for 34 days.
34 days of inflammation. That's a marathon for an in vitro experiment.
It is. They really wanted to simulate a chronic state, and the results were pretty striking.
What happened to the heart tissue?
The chronic inflammation clearly triggered hallmarks of arrhythmia, specifically in the atrial cardiomyocytes, ACMs, the heart cells of the upper chambers where AF originates. They started seeing spontaneous, irregular rhythms, faster beat rates.
Okay, regular beating, faster race. But did they see the specific electrical problems that cause AF in patients?
They did, and this is crucial for validating the model. They documented specific arrhythmogenic events. Early after depolarizations, EADs, and delayed after depolarizations, DADs, plus spontaneous calcium elevations, SCAs.
Okay, EADs, dead, SCASy. Can you quickly break down for us why those particular glitches are the smoking guns for AF?
Sure, so EADs are like the heart cell trying to fire again before it's fully recharged from the last beat. DADs happen after it's recharged, but they trigger an unwanted extra beat. Both mess up the timing.
Got it. Extra beats throwing off the rhythm.
Exactly. And the SCAs, those spontaneous calcium bursts, add to the electrical instability. When you have EADs, DADs, and calcium instability all happening, especially in the atrium, it creates the perfect storm for that chaotic, irregular quivering beat that defines AF. Seeing these specific events means the hHMA is functionally replicating the pathology.
Okay, the model gets the pathology right. What about the mechanism? What was driving these electrical problems under inflammation?
They dug into the molecular pathways. The inflammation treatment caused a dose-dependent increase in genes linked to AF, and one pathway really stood out, the NLRP3 inflammasome.
The inflammasome, we hear that word a lot in inflammation research.
We do. It's a protein complex inside cells that acts like a danger sensor. When activated, it triggers a powerful inflammatory response, including the release of IL-1. And here, they saw upregulation of NLRP3 itself, plus CASP1 and IL-1B, all key components of this pathway.
And they could actually see this happening.
Yes. Using immunofluorescence staining, they saw increased levels of NLRP3 protein and also these things called ASC specs. ASC specs are basically visual confirmation that the inflammasome complex has assembled and is active. And importantly, they saw these primarily in the atrial cardiomyocytes, the ACMs.
So the inflammation directly triggers NLRP3 activation in the atrial cells, and that drives the arrhythmia, a clear mechanistic link in this human model.
Precisely. It strongly points to chronic NLRP3 inflammasome activation as a core driver of these AF-like events in response to inflammation.
Which leads us right back to our resident immune cells. What were the TRMPs doing during all this? Were they just innocent bystanders caught in the inflammatory crossfire, or were they contributing?
They were definitely not innocent bystanders. They were active participants and, unfortunately, seemed to be fanning the flames.
How so?
When the researchers isolated the TRMPs from these inflamed hHMAs and analyzed them, they found the TRMPs themselves had shifted into a distinct pro-inflammatory phenotype. Meaning they started churning out inflammatory signals. They upregulated the NLRP3 inflammasome genes themselves, NLRP3, CSP1, IL-1B, and also other pro-inflammatory cytokines like IL-6 and TNF. At the same time, they downregulated genes typically associated with anti-inflammatory or repair functions like VEGF-A.
So they basically switched sides from builders and maintainers to inflammatory amplifiers.
It looks that way. Although, interestingly, there was one nuance.
Oh.
The source notes also mentioned they saw an increase in IL-10 expression in these TRMPs, and IL-10 is usually considered an anti-inflammatory cytokine.
Wait, so they're pumping up pro-inflammatory stuff and anti-inflammatory IL-10 at the same time. What's that about?
It's actually quite realistic. It suggests that even as the TRMPs are contributing to the overall inflammation, they might also be trying, perhaps futilely, to put on the brakes simultaneously. This kind of complex, mixed signaling is something we often see with macrophage responses in vivo. It adds another layer of complexity and realism to the model.
Okay, so a complex response, but the net effect. Did having TRMPs on board make the AF worse compared to organoids without them?
That was the critical comparison. They took standard HHOs, no TRMPs, and the hHMAs with TRMPs, and hit them both with the same medium dose of the inflammatory cocktail.
Yeah.
Both models developed arrhythmias, as expected, but, and this is the key point, the hHMAs, the ones with TRMPs, showed a significantly higher atrial beat rate compared to the HHOs without TRMPs.
Higher beat rate, meaning more excitable, more prone to the chaotic rhythm.
Exactly. The conclusion was pretty clear. Under these chronic inflammatory conditions, the resident TRMPs actually accentuate atrial excitability. They make the arrhythmia worse, they accelerate it.
Wow, that's a finding you simply couldn't get without having those resident immune cells integrated into the model. It directly implicates them in the severity of inflammation-driven AF.
Absolutely. It highlights the power of this more complete model system.
And this finding naturally leads to a therapeutic question. If TRMPs are making it worse, and the NLRP3 inflammasome is driving it, can you target that pathway to fix it?
That's what they tested. They performed a rescue experiment. They took the inflamed hHMAs, the ones showing arrhythmias, and treated them with MCC950.
MCC950, what does that do?
It's a well-known specific small molecule inhibitor of the NLRP3 inflammasome. It basically blocks its activation.
And did the rescue work?
It did. Treatment with MCC950 significantly calmed things down. It reduced the spontaneous irregular rhythms and also cleared up those ASC specs, the visual marker of inflammasome activation.
So blocking NLRP3 reversed the AF phenotype in this human model. That's pretty strong validation.
It absolutely is. It validates the mechanism they identified, and it also demonstrates the potential of the hHMA platform for testing potential drugs, for preclinical screening of antiarrhythmic therapies that target inflammation.
Okay, this has been a really fascinating journey into the heart's ecosystem. Let's try to bring it all together for the listener. What's the main takeaway from this deep dive into the hHMA?
I think the core message is the dual utility of this platform. It's a major step forward for heart research on two fronts.
All right, front number one.
It's an incredible tool for understanding basic development. It gives us the first really detailed look in a human in vitro setting at what these essential TRMPs actually do their roles as the heart's integrated electrician, cleanup crew, and engineer. Functions we suspected but couldn't really dissect in humans before.
And front number two.
It's a powerful new disease model. Specifically for inflammation-driven atrial fibrillation, it recapitulates key features of the disease, including the specific electrical defects, and importantly, implicates the NLRP3 inflammasome and the TRMPs themselves in driving the pathology.
But as always with models, we need to keep the limitations in mind, right? The sources are careful to point this out.
Absolutely essential. Context is key. These hHMAs, as advanced as they are still in vitro systems. They're grown in a dish.
Meaning they
lack several things present in a real human heart. There's no functional blood supply, no vasculature pumping blood through them. They don't experience the mechanical forces, the stretch and strain of a beating heart inside a chest.
And the immune system is still simplified, right?
Right. They have the resident macrophages, which is huge, but they're missing the full systemic immune context, things like circulating T cells, B cells, neutrophils, which all interact with the heart in disease states in vivo. So translation to humans still requires caution.
Okay, limitations noted. But even with those caveats, the data here is really provocative. We see TRMPs accelerating AF in response to inflammation, and we see that blocking the NLRP3 inflammasome can rescue it in this human tissue model. So if the fundamental problem involves these resident immune cells in the atria getting activated, polarizing, and secreting inflammatory stuff like IL-1 that directly messes with the heart's electricity, what does this suggest for the future?
Well, it really pushes us to think differently about treating AF, doesn't it? Especially AF linked to chronic inflammatory conditions, which is increasingly common.
How so?
It raises a really important question for the future. Could we move beyond just managing the symptoms of AF, controlling the rate or rhythm, ablating faulty circuits, and start targeting the underlying immune drivers?
You mean immunotherapies for heart rhythm disorders?
Potentially, yes. Could therapies targeting specific macrophage activities or perhaps targeting the NLRP3 inflammasome pathway directly become a way to treat or even prevent AF in patients with systemic inflammation? That's the kind of future direction this advanced modeling work opens up. Something for you, the listener, to really think about. Are we on the cusp of treating AF not just as an electrical problem, but as an immune problem too?